So we have one last speaker, which is waiting for us. Alexandra, tu es là? Alexandra.
Oui, est-ce que vous m'entendez?
Oui, très bien. C'est super. Alors, merci Alexandra. Tu veux le présenter? Voilà. Donc, Alexandra Henrion Caude est vraiment une figure française dans la génétique. Elle a, comme nous tous, un peu eu des ennuis à parler dans la vérité, dans la science, disons pas la vérité, mais la science, et d'expliquer les choses. Et on est très contente, Alexandra, que tu as pu te libérer. Elle devait être là, mais pour des raisons familiales, tu n'as pas pu être là. Mais on est très content. Tu vas parler donc de la santé dans le génome et dans le vaccin. Je vais le faire aussi en anglais. So Dr. Alexandra Henrion Caude is geneticist. She's very known in France because she also had the Eisenhower Fellow, which is a prestigious prize. Very few French people have it, and it was something that she will explain maybe that to facilitate exchange of science and culture. It is a very prestigious thing. And she has created, and she's the director and research director of Simplissima, which is doing an international research institute. You can explain, Alexandra, I don't want to say. So please, thank you for being there, and you have a very important and touchy subject. We look forward.
Thank you so much, Astrid. I'm not sure whether I should do it in French or in English. It is true that the habit for us all is to proceed in English. I really strongly thank you for putting up such a fantastic and awesome program that enables us to speak science, to speak whatever our field of expertise is in a free manner. And I have to say that I miss quite a lot those exchanges in a free manner, where other scientists challenges you and you have this ping pong thing. So just to give a little overview on what I'm doing with Simplissima Research Institute is actually very timely. It was, the idea was about finding simple solutions, simple health solutions that could be ethical, durable, low-cost solutions. So basically what we have seen that we are missing currently, going to the most complicated strategies or the less appropriate or the less ethical ones. So to start with my presentation, I will share my screen, but I'm unable to share my screen. If I can get the hand on it. Excuse me for this technical issue. Share screen. All right, there we go. How is it? You see well?
It's good. You can go ahead.
It's good. All right. Okay. So so to start with, I'll I'll tell you what we do normally in science. In science, we have this huge gap that you see on the photograph, and we are crazy enough as to think that we can fill the gap with such a bridge, and the bridge being of knowledge. And obviously, it is a very tough challenge that we are all taking when doing science. And to do so, we are coming from taking what we have, so the material, what I will call common science, and we aim at bridging the gap through ideally great discoveries that are breakthrough, and a number of breakthrough make the path to knowledge. In order to reach the spirit of extraordinary science. And for the spirit, to me, is the inspiration of the theory that we have in science, or the laws that we put up in science, meaning that those theory or those laws, the laws are a common ground, but the theory are like a ceiling that one wants to break at one stage, because we all know that it's a theory and that we don't have the proof for it. Einstein put up a theory, Darwin put up a theory, and those are very inspiring at a time of history. And the idea is really to be building the bridge meanwhile to this, maybe to this theory or to other theory. That's the little, and so obviously there's no such thing as a pure scientific consensus. And I say that because in France, the decision of our government was to put up an organization now of a consensus scientific point of view in reaction to what has happened, meaning the people like me who have uttered a voice that was a bit different from other voices that were all the time heard. Organization now of I have a jet lag point of view in reaction to what has happened. How can I do that? Meaning the people like me who have uttered a voice that was a bit different from other voices. It seems to me that there is a big lag between what I say and what I hear. I hear a sound back. How can I do that? Is there a way I don't have an echo? It seems to me that there is a big lag between what I say and what I hear. I hear a sound back. Can we fix that? Is there a way I can fix it?
Voice now in the back?
Yeah, I hear a sound in the back, so an echo that is really far from what I'm saying, so it's very disturbing.
C'est ça?
Non, quand je t'entends, c'est parfait, mais quand je parle, il y a un espèce d'écho de moi, de ma propre voix, avec un énorme décalage.
C'est le retour.
Mais pas quand on est en...
Ouais,
quand on
tu
est
as
en
le
réel,
premier
comme
zoom, alors c'est peut-être pour ça. Hein?
Du coup, je sais pas.
Attends 2 secondes, ils sont en train de regarder la table mixage.
Qu'est-ce qu'on fait? C'est juste pour savoir.
Il est là.
C'est bon? Je continue?
Oui, tu peux continuer. Non, t'entends bien.
Bon, OK. Alors, so COVID-19, there is obviously a lot to say, but what I wish to say is that on COVID-19, there had been a schisma of misunderstandings, and these misunderstandings is based on the fact that we have a narrative that is made out of gaps. So as you saw, my consideration of science and the way we go little by little, step by step, of course, when facing a new crisis, we have a lot of unknown. The idea is not to hide this unknown and to keep it in transparency to the people. And I believe that there has been some trouble here because some scientists were under a narrative of understanding and some scientists were on another narrative. And so there is no such thing as conspirationist or whatever in science. We're just blind people trying to make up, to bring some light to something that we don't know. So what I'm saying is actually very heavy in terms of consequences. Let's see what we did with this COVID-19 crisis. We redefine the word pandemics. Indeed, we as scientists were under this understanding that pandemic had the severity component in it, meaning that it had to be not only a contagious disease, but also it had to be severe in terms of lethality rate. And this is something that one can discuss as of today. And the issue came by the fact that the World Health Organization in 2009 had a definition which evolved with the time. And obviously, if we were coming back to this definition, which is, in fact, it was the fact that a new virus emerges in humans, a minimal or no population immunity was observed, and it caused serious illnesses, high morbidity, high mortality, and spread easily from person to person. So we see that we do have a number of criteria met here. But the fact is that today we revised the lethality rate to a percentage that raises seriously the question of causes serious illness and high morbidity mortality, because we have reached a percentage that is similar to flu. So typically, if we revise the definition as of today, are we still in a health emergency case? And do we still proceed with all these things that we are doing at the moment? So to me, and for the lawyers and for the people who want to make a change to this the situation, which is somewhat desperate for a great number of people, we have to start to this point. This point is completely critical. And if we have a revised comprehension on the status of the pandemic as of today, we have the key, to me, to a great number of issues that we're facing. The number two is the RT-PCR that was wrongly used as a diagnostic test, when we perfectly know that it shouldn't have been done that way, because RT-PCR should have been at least a step to sequence. And had we done the RT-PCR and then sequenced, we could have followed what was going on, the different strains, and the fact that we were really dealing and facing cases of SARS-CoV-2 COVID-19 patients. This is something critical that the Portugal court has actually pointed out as being a wrong test, and there is a number of statements as well through the CDC and at different stages that it was wrong to use it as a diagnostic test. This is again the basis of a number of decisions. Redefinition of the treatments. Obviously, we have a number of doctors around the world had put up with the observation that macrolides were a great solution, hydroxychloroquine, ivermectin, and the thing is that because they were redefined as not to be used, forbidden, very, very wrong, we are facing this misunderstanding again. Why do I put up a vaccine? Because again, the vaccines are being redefined. They are not vaccine as we currently have been using vaccine or developing vaccines, and those vaccines are different. The redefinition is also based on the strategy. The strategy says that you have to wear masks, but then if masks are efficient, why do we have to put up with a distance that the French government has asked to be 6 meters 50 centimeters now, as of today? And so if mask and distance are efficient, why do we go to confinement? And if mask and distance and confinement are efficient, why do we now go to vaccine strategy? So we see here that obviously there is something wrong again in this whole dynamic. The last thing that I wanted to raise as to the misunderstandings, but there are way more misunderstandings, is the evolving parameters to assess the crisis. So we keep on changing the parameters, and so we are not talking the same language. And so, of course, scientists have to disagree, because we are, to me, typically, a good parameter to assess the severity of an epidemic is the excess of mortality. Apparently, no one is using this parameter, which is on a society what one wish to aim and to assess. And another parameter that we have in France put up as being a very potent parameter to be in the anticipation, but yet which is not used as a consensus parameter, is the dosage in wasted water. And in wasted water, you can do the assessment of the presence of the RNA of SARS-CoV-2 and be in anticipation of the patients in a particular area. So this is a fantastic, innovative tool, but I don't know why people are not using it, because it seems really robust. Okay, I will go on because I have a lot to say. And I will move on to the part which is important, is the fact that those vaccines are so-called vaccines. I prefer to say those genetically engineered experimental vaccines, because this is exactly what they are dealing with our genetics. And this is very important, because one has to understand and has to have an updated view on what is our current knowledge on genome, on our heredity, and not in old view that messenger RNA is something very static and very ideal, that it does exactly what you want it to do. What is heredity? Heredity is no longer only based on genetics, but it is based also on epigenetics. What is epigenetics? Genetics people have now understood what we are talking about. It is this idea that those two honeybees, one is a queen on the left side, the other one is a worker on the right side. And they have the same DNA, meaning they have the same genetics. Yet you do see that their morphology is rather different, even like the abdomen doesn't have the same stripes, the size, the behavior, and the organs are not the same. So what is it? Is it that heredity is only linked to genetics? No, it means that there is something else. And this something else, interestingly enough, is about the epigenetics. So, which makes those bees look so different, the queen and the worker, and behave so differently, is the epigenetic, which is basically the changes that the environment brings to your genomic information altogether. So we are talking of DNA, of RNA, and even now of recently, since the discovery of circulating factors that all influence your epigenetics. So the human genome is not only into your nucleus, but it is also in your mitochondria. And so just to give you an overview of what is the human genome in the nucleus, it is a rather complex thing, as you can see. And when we say that we know everything about your genes, we're always talking of this red rectangle at the very right of 1% of protein coding sequences. Those are your so-called genes. All the rest is basically the unknown, basically the fantastic fluidity of our complexity. And it is composed, and I should stress the fact that 8 to 9% of our genome is composed of viral sequences. So in fact, we interact all the time with bacteria, with viruses, and we get this interaction that we even put up in our genome some sequences from the viruses. So this so-called junk DNA, which was the part that is not the 1% tip of the iceberg, the red one, is very interesting because it has, this is a plot that was from Taft and Matic in 2007. And they plotted the different species, prokaryotes, simple eukaryotes, vertebrates, to human on the very extreme right, the red extreme bar. And in fact, what did they do? They didn't plot according to an increasing complexity as it looks like. They just plotted on the ratio of non-coding DNA to your total DNA. Meaning that in this fantastic load of these unknown sequences, you have the complexity of the living that goes from prokaryotes to simple eukaryotes to fungi to plants to vertebrates. So this is absolutely fantastic, and it should bring one conclusion for us, which is that we have to be really humble as to our understanding of when we do anything with our genome. The second thing that I want to share is that the cartoon saying that you know, a gene is a gene is much more complex than that. So now we are talking of the 1% that you saw, the 1%, but even this 1% is tremendously much more complex than we present it. And in fact, I don't know if you know the millefeuille cake in France, but to me, this is a millefeuille. So when you think this is a gene, within the gene, you have a great number of RNA sequences that are embedded into your gene and that actually are very important in the cell to bring a flux of information. So when we talk of mRNA, we talk of all these information that are embedded in the sequence. Now, what do we know more on RNA biology? Is that RNA biology brings us a number of information as to how this RNA can work. And this RNA can work in the sense that you have possibility that they will induce chromatin remodeling and histone modifications. So through different interactions, you have an interaction RNA-RNA, and that brings a lot of cascades of regulation. You have interaction with proteins, and you have this idea that they actually are precursors for other small non-coding RNA. How can we see those different little RNA? They are like a brake to your Ferrari. If you alter one of these cable of your brake, you think that you're safe because your Ferrari can still be used. Those are those cables are actually the way those microRNA function. They bring a negative, they bring the power of inhibition to your system, and they can be of viral origin or they can be of your own origins. And if you cut one of these cable and you face that a child is crossing the road, Now you want to brake and you don't have full power on your brake. And this is the reason why a number of consequences may not be seen immediately, but may be seen in the long term, because you have to face the situation where you really needed this information of your microRNA, for instance. Another aspect that I want to bring to your attention is that the common assumption was that DNA was copied to RNA and everything was simple. But actually you have more than that, because your RNA can get edited. And this is known since 1986 and has been more improved in our knowledge that other transversions could be made onto this RNA distinction in 2014 and in 2016. So one has to understand that the RNA sequence is not something completely stable, and that it can be edited in a great number of manners that we still don't know of. We know it exists, they exist, but we don't know how they are turned on. Given this fantastic knowledge that I think completely revolutionizes our understanding of genome, our understanding of heredity, we are putting up a rushed development of these experimental procedures to put up vaccines, genetically engineered vaccine at a worldwide scale. This is the worry. This is the concern. And the concern is that through this rush development, any serious adverse events, instead of being observed during 24 months, as it is normally what is taking place, it has been cut down to 6 months. Within 6 months, they will be observing the serious adverse events with a rushed experimental procedure. So we see that something is going in the wrong direction. We immediately see that we have to put a halt as a scientist because we have this knowledge. We have to share what we know, and we have to say this: the path we are taking is a dangerous one. Most, if not all, strategies of vaccine that are summarized in this table have never been used in humans, and most of the time either never been tested in animals. This is one serious concern again, is that not only they are rushed, but also they were never used and never really fully experienced or tested in animals. You have different types of vaccines. You have the GMO vaccines, you have the mRNA vaccines, you have the DNA vaccines, and you have some specificity in different recombinant, different nanoparticles of vaccines, using different, even plant cells, a wide variety of many parameters that are completely new. So to summarize, I don't know what the timing, Astrid, because I didn't look at the time, I should have been looking.
Yeah, it's okay. Combien de temps? Huh? Yeah, you have 5-10 minutes.
Okay, great. So yeah, thank you. Yeah. So I have summarized here some of the current challenges that we are facing. The first challenges that I see is the indication of a public health policy in regards to COVID-19 vaccines. Given what Pascal has presented, what the other doctor has presented, we have a number of treatments that exist that give very good results, and and therefore, given this aspect, and given the fact that we have had other epidemics such as H1N1 or other epidemics, and we have realized after the epidemics was gone that the strategy of a public health policy of vaccination was not the solution, we should be learning from our past experience. And therefore, we should strongly raise the indication of a public health policy of anti-COVID-19 vaccine strategy. Meaning, I think the fact of developing a vaccine on a solution is a solution that should be undertaken with an indication. Typically, it seems to me that the severe cases of COVID-19 are an issue that we still haven't really well solved, and therefore we should have typically focused on those severe cases. It has not been planned that way. No indication was discussed to start with. No discussion as to why we should put up a worldwide strategy of vaccination had any rationale. This is a discussion that has to occur, and this is likely the reason why a great number of people, let's say more than half, at least in France, don't want to take the vaccines. I think because they didn't have the debate. They just don't understand why everyone should go and get vaccinated. And indeed, we need to have the debate of stating, of understanding the education of a public health policy of vaccination, if there is such, or if it is not, it's more a vaccination for a certain category of people. This is number one. Number two, are the people who are currently getting the vaccines informed that they are currently being enrolled in a biomedical protocol? This is a question I raise, and the reason being that the clinical trials are still ongoing. The clinical trials of all the laboratories that I've showed to you on the different strategies are ongoing. Yet, people are being vaccinated, and to the best of my understanding, they are not informed that they are being enrolled in a biomedical protocol. There is a strong ethical discussion to have here. Another discussion, informing people about the specific risks with anti-coronaviruses. This is very well explained on this article on the right-hand side that was recently published and stating from Cardozo and Vesey from New York, who are saying, informed consent disclosure to vaccine trial subjects of risk of COVID-19 vaccines worsening clinical disease. And what they are basically saying in this article is that we need, given our past experience on anti-coronavirus vaccination, we need to specifically inform the patient that the risks are that it will worsen the clinical disease. How does it do that? In fact, it is based on a phenomenon which is called antibody-dependent enhancement. And this antibody-dependent enhancement makes that, yes, you may have been producing antibodies, but those antibodies, instead of being neutralizing, in fact, instead of neutralizing the disease, will actually have the reverse effect of facilitating the disease, of facilitating the entry of the virus into your cell, of facilitating the replication sometimes of the viruses. This is a very big concern, which explains to you why there has never been any approved vaccination on any coronavirus, on any SARS-1, on any MERS, and this is something that should be raised to the people. The other issue I have is we can have, not I have personally, but we should be having, is the fact that the targeted population are actually never defined as groups of study in the trial assays, in the clinical trials. This is a concern that made Dr. Doshi to put up this article in British, he's associated editor of British Medical Journal. And he currently, he asked the question, will COVID-19 vaccine save lives? Which is the question one should be asking. Current trials, the answer is current trials aren't designed to tell us. So this is once more a very, very big concern that we should be having. Oops, I did a wrong thing. Sorry about that. Yep, coming back. So the targeted population is not defined as group of study, and the targeted population, for instance, would be the people at risk, and they were not defined as a group of study on which we would specifically assess the vaccination. Other aspect, the gene modification, yes or no? We, I don't know in other countries, but in France, it seems to be a total consensus. Those vaccines are not going to change your, to bring any gene modification. So I wonder what we are talking about. And this is again a big misunderstanding, coming back to the root of the basis. We are talking typically of mRNA vaccine, Pfizer, BioNTech, or Moderna strategy. Those mRNA vaccines, those genetically engineered experimental vaccines are designed for you to, for human to start producing a protein that they never produced before. So this is not gene modification. So I have to say that I am not playing the fool, but I'm just trying to understand how can these people be saying that it is not modifying your genome when it is actually bringing an information to your body that your body didn't have. Your body was not a producer of an industrial producer of a spike protein of this virus. This is an issue. Now, are there other ways that, beside the fact that you are going to produce a protein that you never produced before, in the hope that maybe it will turn on an immunologic response, and apparently the efficiency as to synthesizing antibody is strong. But now, are these antibodies facilitating antibodies or neutralizing antibodies? Nobody knows. And this comes back to this article from Cardozo that I mentioned about the specific risk of worsening the disease with anti-coronavirus. The modification that mRNA may bring and cannot, in other words, I don't see a way that it cannot have an impact on the epigenetic of your cell. Because as you saw my introduction on putting you all the cartoon of the fantastic fluidity and even changes that RNA bring within the cell, you cannot be in the expectation that this mRNA is not going to have an epigenetic influence on your cells. This is, to me, not reasonable. And this goes completely against all the fantastic improvement that we have made in our knowledge and all the literature that has been ongoing on SARS-CoV-2 and epigenetics and within the last month, which is a huge piece of literature in the field. So epigenetically, I don't see how they cannot be actually changing your, have an impact. And now in terms of changing your DNA, one can have the concern that your cells may have reverse transcriptase because maybe you are HIV, or because maybe you have other viruses that provide you with reverse transcriptase, and your mRNA may become a DNA and may modify your genome. This has to be foreseen as a possibility, and therefore this has to be studied. And unfortunately, I didn't see that genetic parameters, genotoxicity, were in the list of the things that had to be studied. There are other concerns, but I don't want to put them all. This is to give you a glance as to the complexity that we have at the moment. DNA is being transcribed into RNA, RNA is being translated into protein, but you have the possibility of, through other RNAs, and you have the possibility of the DNA to replicate the RNA, of also using ribosome self-amplification, the proteins with the prions of some self-reproduction. And this is one level of complexity, but you have to envisage the fact that this DNA will be modified, and this is called your epigenome. These RNA are modified, and this is your epitranscriptome. And these proteins are being modified, and this is your epiproteome. And all that can be transferred to the next generation. So, oops, gosh, sorry. So to conclude, I will say that a vaccine, well, my question is, how can we be producing, or not producing, but like putting up vaccination policies when vaccines were not made for the severe cases of COVID-19 and were not made to prevent transmission human to human? Can we call them vaccine? And is a genetically engineered vaccine that is completely experimental, can it be called again a vaccine? Or does the word experimental need always to be tied up with the word vaccine, and as well as the genetically engineered, because this is completely new strategies that were never done in human, and that can have a great number of consequences. Innovation is great. And that's why Astrid, all of us scientists are working on. Innovation yet is not always a synonym of progress. Is it a progress to change our consideration of human integrity? Why do I say that? Is this because when we are using mRNA vaccine on healthy individuals, and to me this healthy aspect is really important, we consider human being as a biotech tool to be producing a viral protein. So we are engineered as to being the producer. This is touching our human integrity. This is a critical aspect that one has to really embrace in terms of the revolution that is coming along with this change of human integrity. We are not a bacteria to be producing such and such proteins that we want to be producing. The other aspect is this is changing our genetic information, our heredity information as well. So once again, is that an innovation to be changing our genetic information? I'm asking the question. And is this a progress? It's an innovation for sure, but is it a progress to be changing healthy people genetics, the genetics of healthy people? We are taking the risk of transmission with these vaccines, whether at the intergenerational level or transgenerational level, meaning that maybe the next generation will not have anything, but the following one, yes, because once again, we are touching on epigenetic issues. And nobody can tell me to date that those vaccines, those mRNA, those lipid particles that have the mRNA will not flow through your germ cells, because it has been, when you go into the circulation, when you alter the vascularity of the muscle, at the moment you do so, some particles will go into your circulation and will circulate in a number of cells, whether the brain or organ, whether the brain, whether the germ cells, and no one can say that this cannot be taking place. So we are taking the risk of a modified transmission in a manner that we know nothing of. And a last thing that may sound really ridiculous for the non-scientist, but to me, which is very important, I have been putting together with many colleagues, we have been putting a lot of energy as to the hopes that we're bringing RNA-based therapeutics. I was even an editor of a specific issue on those therapeutic with the hope of treating patients. And what is going on at the moment is terrible for us, because this rushed strategy is in the phase that it may ruin all our knowledge and the promise of RNA-based therapeutics, because it will give a very bad presentation because it is so rushed, because the experience in animals were not proceeded in the right time. And therefore, we may face a situation that the public, will never want to hear about RNA-based therapeutics anymore for diseased people with severe diseases. And this, once more, is a big concern that I have, because what a waste. I thank you very much for your attention, and will be happy to answer to the questions that whenever it comes to.
Thank you, Alexandra. C'est super. It was very, very detailed, and I'm sure this is going to be fantastic to have an English version of the explanation. It will be translated for the French. We will have 2 versions. You want to say something? Yeah.
Voilà, c'était pour remercier le docteur Alexandra Henrion de son intervention, qui va répondre à beaucoup de questions. Donc on a eu pas mal de questions sur Internet, bien sûr par rapport à la vaccination. Je pense qu'on ne pourra pas toutes les proposer. Astrid va pouvoir vous en poser quelques-unes. Et pour les autres auditeurs, ils ne s'inquiètent pas, il y aura la post-production en plusieurs langues et où ils auront pas mal de réponses.